Agricultural College and Research Institute, Kudumiyanmalai. Department of Plant Pathology proudly presents Project Report Preparation and Presentation. Project title: Discovery of Novel Microbes for Plant Growth Promotion. Project Guides: Dr. S. Nakiran, Dean, Agricultural College and Research Institute, Kudumiyanmalai. Dr. S. Paul Sebastian, Associate Professor, Environmental Science. Collection of samples. Sample A, Plant Sample 1, Banthulasi, Croton Bone Plantianus. Pull out the plant from the land. Gently tap the rhizosphere soil adhering to the root zone. Sample B, Plant Sample 2, Asian Spider Plant, Cleome viscosa. Soil from the rhizosphere region was collected. These plants were selected for isolation because they remain green under water stress conditions. Sample C, Termitarium sample. Soil from Termitarium was collected. Sample D, Wasp nest sample. Wasp nest with comb length of 8 cm and diameter 5 cm was selected. Isolation of bacterial isolates from collected samples. Weigh 1 gram of soil sample. Weighed soil sample is then added to 10 ml of sterile distilled water. The test tube is then tapped gently for the microbes in the sample to get blended with the solution. Dilutions were prepared up to 10 power 6 using sterile distilled water. 1 ml of suspension was pipetted out using micropipette from 10 power 1 dilution and was added to 9 ml of sterile distilled water. Subsequently, dilutions were made up to 10 power 6. 10 power 5 dilution was selected for the isolation of fungi and 10 power 6 dilution was selected for the isolation of bacteria. Nutrient agar medium was selected for the isolation of bacteria and potato dextrose agar medium was used for the isolation of fungi. A pinch of streptomycin sulfate was added to potato dextrose agar medium to prevent bacterial contamination. Test tube containing 10 power 5 dilution was used for the isolation of fungi. 1 ml of suspension was taken and was transferred to a disposable sterile Petri plate. The plates were then rotated in clockwise and anti-clockwise direction for the uniform distribution of sample. Test tube containing 10 power 6 dilution was used for the isolation of bacteria. 1 ml of suspension was taken and was then transferred to a disposable sterile Petri plate. The plates were then rotated in the same manner done earlier. 15 ml of molten and cooled nutrient agar medium was poured to a disposable sterile Petri plate. The plates were then rotated in clockwise and anti-clockwise direction for the uniform distribution of media. 15 ml of molten and cooled potato dextrose agar medium was poured to a disposable sterile Petri plate. The plates were then rotated in the same manner done earlier. Plates were then wrapped using thin film and were incubated at room temperature. Isolated bacterial isolates from sample 1, Banthulasi. Bacterial isolates from replication 1, 2 and 3 under 10 power 6 dilution. Bacterial isolates from replication 1 and 2 under 100 microliter dilution. Isolated bacterial isolates from sample 2, Asian spider plant. Bacterial isolates from replication 1 under 10 power 5 and 10 power 6 dilution. Bacterial isolates from replication 3 and 1 under 100 microliter dilution. Isolated bacterial isolates from sample 3, Termitarium. Bacterial isolates from replication 1, 2 and 3 under 10 power 6 dilution. Bacterial isolates from replication 1 and 2 under 100 microliter dilution. Isolated bacterial isolates from sample 4, Wasp nest. Bacterial isolates from replication 1 and 2 under 10 power 6 dilution. Bacterial isolates from replication 1 and 2 under 100 microliter dilution. Subculturing of the bacterial isolates. Colonies with unique characteristic features were used for subculturing. Selected colonies were picked out using an inoculation needle. Each colonies were streaked over the surface of the solidifying medium by simple streak method. 3 replications were maintained for each bacterial colony. The same process is repeated for subculturing other selected bacterial isolates. Subcultured bacterial isolates from termitarium sample. Subcultured bacterial isolates from termitarium sample of replication 1 under 100 microliter dilution. Subcultured bacterial isolates from Asian spider plant sample. Subcultured bacterial isolates from Asian spider plant sample from replication 1 under 100 microliter dilution. Subcultured bacterial isolates from termitarium sample. Subcultured bacterial isolates from replication 1 under 100 microliter dilution. Subcultured bacterial isolates from termitarium sample. Subcultured bacterial isolates from replication 2 under 100 microliter dilution. Subcultured bacterial isolates from Bantholosi sample. Subcultured bacterial isolate from replication 2 under 100 microliter dilution. Subcultured bacterial isolates from wasp nest sample. Subcultured bacterial isolates from replication 2 under 100 microliter dilution. Subcultured bacterial isolates from Bantholosi sample. Subcultured bacterial isolates from replication 1 under 100 microliter dilution. Subcultured bacterial isolates from termitarium sample. Subcultured bacterial isolates from replication 3 under 100 microliter dilution. Subcultured bacterial isolates from wasp nest sample. Subcultured bacterial isolates from replication 1 under 100 microliter dilution. Subcultured bacterial isolate from Asian spider plant sample. Subcultured bacterial isolate from replication 1 under 10 power 6 dilution. Confrontation assay for assessing the antifungal activity of bacteria against Fusarium oxysporum, former species Cubens race 4. Mycelial disc with diameter 8 mm of Fusarium oxysporum, former species Cubens, was made using a sterile cork borer. The mycelial disc was then picked out using a sterile needle and was placed near the peripheral end of the sterile disposable Petri plate. Bacterial isolate was taken out using a sterile bud and was then streaked against pathogen near the peripheral end of the sterile disposable Petri plate. Control untreated with bacterial isolate. Mycelial growth 8.3 cm. Isolates that effectively restricted pathogen growth. Bacterial isolate 1 from Asian spider plant. Mycelial growth of pathogen 5.3 cm. Inhibition zone 4.4 cm. Percent inhibition over control 36.13%. Bacterial isolate 1 from wasp nest sample. Mycelial growth of pathogen 3.9 cm. Inhibition zone 4.4 cm. Percent inhibition over control 53.01%. Bacterial isolate 2 from wasp nest sample. Mycelial growth of pathogen 4.3 cm. Inhibition zone 4 cm. Percent inhibition over control 47.77%. Bacterial isolate 3 from wasp nest sample. Mycelial growth of pathogen 4.6 cm. Inhibition zone 3.7 cm. Percent inhibition over control 45.17%. Bacterial isolate 2 from Asian spider plant sample. Mycelial growth 4.2 cm. Inhibition zone 4.1 cm. Percent inhibition over control 49.15%. Bacterial isolate 1 from termitarium sample. Mycelial growth 5 cm. Inhibition zone 3.3 cm. Percent inhibition over control 39.75%. Bacterial isolate 2 from termitarium sample. Mycelial growth 4.4 cm. Inhibition zone 3.9 cm. Percent inhibition over control 47.59%. Bacterial isolate 3 from termitarium sample. Mycelial growth 3.4 cm. Inhibition zone 4.9 cm. Percent inhibition over control 58.43%. Among all the bacterial isolates observed, it was found that bacterial isolate 3 from termitarium sample, bacterial isolate 1 from wasp nest sample, and bacterial isolate 2 from Asian spider plant sample significantly reduced mycelial growth to a greater extent. Pure cultures of best bacterial isolates. Pure cultures of bacterial isolates that significantly reduced mycelial growth are maintained individually. Preparation of broth inoculated with bacterial cultures. 200 ml of sterilized nutrient broth was taken for inoculation. A loop of bacterial mixture was taken out using a sterilized earbud. It was later inoculated in the sterilized nutrient broth under aseptic condition. The inoculated broths were then placed on a rotary shaker for bacterial multiplication. Broths were placed on the rotary shaker at 150 rpm for 2 to 3 days. Broths inoculated with bacterial isolates from Asian spider plant and wasp nest sample. Broths inoculated with bacteria from Asian spider plant and termitarium sample. Evaluation of antagonistic effect of bacterial isolates under in vivo condition. 8 grams of black gram variety Vamban 8 seeds were weighed and taken. 1 ml of respective bacterial broth was taken, and to it 9 ml of sterile distilled water was added. Seed treatment was done by treating the black gram seeds with this solution. The treated black gram seeds were then allowed for shade drying for about 30 minutes. The shade dried seeds were then used for sowing. Drenching of seed sown plots with bacterial broths. 50 ml of respective bacterial broths were taken and was transferred to a rose can of capacity 5 liter. Water was then added to make up the volume up to 5 liters. Drenching was done once in every week. Efficacy of bacterial isolates on germination percentage and seed vigor index of black gram. The efficacy of each bacterial isolate on germination percentage and seed vigor index of treated black gram seeds was assessed. It was found that seeds treated with broth inoculated with bacteria from wasp nest sample showed significantly higher germination percentage, followed by seeds treated with broth of bacteria from termitarium sample and seeds treated with broth inoculated with bacteria from Asian spider plant sample. It was also found that seeds treated with broth inoculated with bacteria from wasp nest sample showed significantly higher seed vigor index, followed by seeds treated with broth of bacteria from termitarium sample and seeds treated with broth inoculated with bacteria from Asian spider plant. Efficacy of bacterial isolates on root architecture development and root nodule count in black gram. The efficacy of each bacterial isolate on root architecture development and root nodule count in treated black gram seeds was assessed. It was found that treatment 5 of black gram treated with broth inoculated with bacteria from Asian spider plant had good architecture development with the presence of long primary roots with dense lateral fine root hairs. It was also found that treatment 5, treatment 2 and treatment 3 treated with broths inoculated with bacteria from Asian spider plant, wasp nest and termitarium sample respectively had high root nodule count. Efficacy of bacterial isolates on plant growth promoters of black gram under in vivo condition. Control plot untreated with bacterial broths. Plant height 17.05 cm, leaf length 4.72 cm, leaf width 3.02 cm, number of leaves 12, leaf area index 0.372. Treatment 1 treated with broth of bacteria 1 from Asian spider plant sample. Plant height 17.33 cm, leaf width 5 cm, leaf width 3.27 cm, number of leaves 18, leaf area index 0.711. Treatment 2 treated with broth of bacteria 1 from wasp nest sample. Plant height 22.71 cm, leaf length 5.63 cm, leaf width 3.81 cm, number of leaves 18, leaf area index 1.033. Treatment 3 treated with broth of bacteria 2 from wasp nest sample. Plant height 21.64 cm, leaf length 5.77 cm, leaf width 3.6 cm, number of leaves 17, leaf area index 0.793. Treatment 4 treated with broth of bacteria 3 from wasp nest sample. Plant height 20.84 cm, leaf length 5.42 cm, leaf width 3.8 cm, number of leaves 17, leaf area index 0.811. Treatment 5. Treated with broth of bacteria 2 from Asian spider plant. Plant height 24.44 cm, leaf length 6.39 cm, leaf width 4.74 cm, number of leaves 17, leaf area index 1.22. Treatment 6. Treated with broth of bacteria 1 from termitarium sample. Plant height 21.16 cm, leaf length 5.48 cm, leaf width 3.7 cm, number of leaves 15, leaf area index 0.679. Treatment 7. Treated with broth of bacteria 2 from termitarium sample. Plant height 20.36 cm, leaf length 5.79 cm, leaf width 3.72 cm, number of leaves 16, leaf area index 0.739. Treatment 8. Treated with broth of bacteria 3 from termitarium sample. Plant height 23.49 cm, leaf length 6 cm, leaf width 4.19 cm, number of leaves 15, leaf area index 0.828. Discussion. In this study, 45 bacterial colonies were isolated from plant samples, Bantholosi, Asian spider plant, wasp nest, and termitarium. Treatment 8. Treated with bacteria 3 of termitarium. Treatment 2. Treated with bacteria 1 from wasp nest, and treatment 5. Treated with bacteria 2 from Asian spider plant inhibited the mycelial growth of test pathogen Fusarium oxysporum former species cubans significantly. Various plant growth parameters such as plant height, leaf length, leaf width, number of leaves, root nodules were evaluated for the treatments and control. On compiling all the results, it was found that treatment 2 treated with bacteria 1 from wasp nest, treatment 5 treated with bacteria 2 from Asian spider plant, and treatment 8 treated with bacteria 3 from termitarium showed significant results in all the parameters that were considered for evaluation.